Okay, I think that ends that video. Okay, so I think we could start this plenary session number 3. I'm Edsel Peña. I'm a professor at the University of South Carolina, and I have been here in the US for quite some time. It's my pleasure to host this session. And we will have 3 speakers later, and this is on health, food, and nutrition. But before that, we have a very important event, which is the conferment of the co-award in science. And we will start that by having our board of directors chair, Dr. Carlito Librela, to give a short, brief introduction, and then a video presentation, and then the conferment of the award. Carlito?
Here.
Starting?
Not yet. It's just the preliminaries.
Yes, I'd like to welcome everyone to the highlight of the conference, which is the presentation of the co-award for science.
I think it's
this is the ultimate award that's awarded, that's provided by PAASE, and well, I just like to welcome everyone and look forward to today's speaker.
And he is one of the speakers later too. Yeah, Carlito. Yeah. So, Albin, would you like to play the video presentation?
Richmond, please. Thank you. Danilo Angeles Tagle, the 2020 Severino and Co. Lectureship Award in Science. Dan was born and raised in Quezon City, Philippines. His parents hailed from Cavite and Bataan. Second in the brood of 7, his family migrated to North America in the early 80s in Detroit, Michigan. There he earned his bachelor's degree in biological sciences and an MS in population genetics at the Wayne State University. In 1990, he received his PhD in molecular biology and genetics at the WSU School of Medicine under the mentorship of the renowned molecular evolutionist, Dr. Morris Goodman. He was also a postdoctoral fellow in human genetics under the guidance of Dr. Francis S. Collins, a world-class geneticist at the University of Michigan. In 1993, he was recruited to join the NIH as an investigator and section head of molecular neurogenetics at the National Human Genome Research Institute as part of the Human Genome Project. His laboratory was involved in the highly collaborative efforts towards the position of cloning of genes for Huntington's disease, ataxia telangiectasia, and Niemann-Pick type C disease. He then became program director for neurogenetics in 2001 at the National Institute of Neurological Disorders and Stroke, where he developed research programs in inherited brain disorders. In 2011, he joined the newly established National Center for Advancing Translational Sciences, or NCATS, as Associate Director for Special Initiatives. He develops catalytic and innovative solutions to scientific challenges and blazes new fields of research that includes the NIH Microphysiological Systems ACA Tissue Chip Program. These cutting-edge and transformative activities involve coordination with other NIH institutes and centers, as well as partnerships with other government agencies such as FDA, DARPA, NASA, and the private sector. Dan has served as chair of the Scientific Advisory Committee of the Philippine Genome Center, also as NCATS Advisory Council Executive Secretary and member Bureau's Acceleration Network Review Board. He was on the editorial board of the journal Gene, as well as International Journal of Biotechnology. With more than 150 scientific publications, H-index of 35, he has garnered numerous awards and patents, including the prestigious 2019 Roscoe O'Brady Award for Innovation Accomplishment and the Henry J. Hendrich Award for Innovative Medicine. Central to Dan's accomplishments and goals in developing disruptive technologies is engaging key resources and expertise through partnerships with various stakeholders in biomedical research.
So we're going to proceed with the conferment of the award, Alvin.
Yes, Giselle will read the certificate of recognition. Giselle, please.
Good morning, ladies and gentlemen. First of all, before the conferment, I would like us to thank again Dr. Severino&Paz for bestowing this annual award to outstanding scientists and engineers in PAASE. So Dr. Severino is one of the founding fathers of the PAASE in 1980, and he was an eminent professor emeritus of engineering. So on behalf of PAASE, we would like to thank the family of Dr. Severino&Paz. On behalf of the PAASE and the PAASE board, I am deeply honored to present this certificate of award to Dr. Danilo A. Tagle, PhD, the 2020 Severino&Paz Lectureship Award in Science, given this day, August 4, 2020, during the 40th PAASE anniversary and 2020 APAMS Manila, Philippines. Signed, Gisela P. Concepcion, President, 2020. Alvin Culaba, Chair, PAASE Committee on Severino&Paz Lectureship Awards 2020, attested by Lourdes Harold, PhD, PAASE President, PAASE Secretary 2020. Congratulations, Dan, and here is your certificate.
Thank you, Giselle.
Where is Dan receiving? Wow, thank you.
Very-
And Dan, here's your $1,000 check. I can keep it if you want. Here.
Thank you, Alvin.
Thank you and congratulations.
Thank you so much.
Congratulations, Dan.
Congratulations, Dan.
Congratulations, Dan. Very well deserved. And you're now a member of the select group of PAASE members that have received the co-awards. And the three speakers in this session are actually all co-research, co-awardees, just like you. So that's really wonderful. So we're going to start with the talks now. And let me share my screen for my introductions of the speakers. See. Okay, so before I introduce the first speaker, which will be Dr. Tagle, our new awardee, let me just mention some of the ground rules for our session, because there's a lot of feedback sometimes. We're going to switch off all our microphones except for the speaker. And then you could ask questions, and you could type that in the chat box. Or if you want, you could raise your hand later, and we could allow you to ask your questions through video or in person. There will be a question and answer session after Dr. Tagle's talk. And then the next one will be after Dr. Pedro Jose and Dr. Carlito Librella's talks. So we're going to start. Let me just introduce a little bit more Dan. Okay, of course, Albin presented a very nice video about him, about his life, and I think I'm going to repeat some of them here. Now, I really like this picture of Dan here. On the left side, he's in front of the famous monument, and I think that's Luneta Park, and that must be Jose Rizal in that monument. But if you look at the picture on the right, Dan really looks like Jose Rizal. So, and the first time he sent me this picture, I said, I know this guy. And it turns out that I was remembering Jose Rizal. So Dr. Tagle has very much in relation with our national hero, which is Jose Rizal. So it's nice to be young and have good hair, and there you could see the resemblance with our national hero. Dan? Okay, so I think Albin had mentioned this, but just to go over a little bit about Dr. Tagle's educational and professional backgrounds. In high school, he was in Letran. I remember my classmates in Philippine Science. There were some of them who went to Letran, aside from, of course, UP and Ateneo. And then he studied at the University of Santo Tomas. But before he completed his degree in Santo Tomas, his family immigrated to the US. And so he completed his degree, bachelor's degree in biology in Wayne State University in Detroit. And then he also completed his master's degree at Wayne State University. And then he proceeded to pursue his PhD in the same university, and he completed his PhD in molecular biology and genetics in 1990. Then he went to Ann Arbor, Michigan, which is, Ann Arbor is about 20 to 30 miles from Detroit. So this must be very convenient for him because he took an NIH NRSA postdoc at the University of Michigan. Now, this is a very prestigious postdoctoral fellowship. Not very many people gets this, and you have to have an excellent record to get this particular award. He worked under Francis Collins, and for those who are following this pandemic, some of you might have noticed that Francis Collins gets interviewed by CNN because he's the current director of NIH. So Dan worked with the current director of NIH, who is now, of course, providing support to Dr. Fauci in this pandemic. As Albin had mentioned earlier, Dan had served in the editorial boards of GENE in the International Journal of Biotechnology. He has published many papers, at least 150 papers, and his AIDS index is 35 when I look at it. And he had gotten numerous awards and patents as well. Now, Dan was a program director of the NIMS, which is the Neurogenetics at the National Institutes of Neurological Disorders and Stroke, prior to his current position, which is now as Associate Director for Special Initiatives at the National Center for Advancing Translational Sciences at NIH. This center is actually dealing with and is responsible for scientific and programmatic coordination to several trans-NIH programs, such as DOD's DARPA. And some of you might know a little bit about DARPA. That is the center in the Pentagon that develops a lot of new technologies, and Dan gets involved in coordinating things there with NIH. So, and it's my pleasure, and of course, congratulations to Dan for winning or for getting the Co-Science Award. And it's now my pleasure to introduce him to give his talk, which is on developing disruptive technologies through team science. And Dan, you you could present your talk now. Let me unshare.
Thank you so much, Edsel. All right. Well, maraming salamat, Giselle and Alvin, certainly for organizing this month-long virtual PAASE conference and for acting as our virtual host. Thank you, Edsel, for the warm welcome. Good morning to our kababayans tuning in from the Philippines, and a pleasant evening to colleagues and friends in the United States. It is indeed a great honor to receive the Co-Lectureship Award in Science. I would like to express my sincere gratitude to Drs. Concepcion, to Dr. Pena, Librilla, and Harold, who are officers of PAASE, and certainly would like to extend my gratitude to the Co-Lectureship Awards Committee, Alvin, Diana, and Seville, for selecting me as this year's recipient. I would be very remiss if I did not acknowledge the previous award recipients in science and engineering and PAASE. These men and women truly represent the best minds in science, engineering, and PAASE. I am truly standing in the shoulder of giants. In particular, I would like to acknowledge the 2008 awardees, Ed Padlan, who actually nominated me many years back for PAASE membership, and to Romel Gomez, who actually nominated me for this award. It is indeed a privilege to share this plenary session also with previous award winners, Pedro Jose and Carlito Leprila. In my presentation on developing disruptive technologies through team science, what I hope to convey to you and cover within the next half an hour or so would be to go over the challenges in drug discovery and development, the need for disruptive technologies, such as the microphysiological systems or tissue chips, using a team science approach through partnerships within the NIH Tissue Chips Consortium, and how you build confidence, how you commercialize, leading to adoption of this technology in development. And I'd like to end with Astra, which is Latin for aim for the stars. And I hope it will become evident why this is towards the end of my talk. For those of you who might sneak out for morning coffee or an evening snack, my take-home message is this: team science is a key enabler to tackle complex challenges and achieve innovative solutions. It involves active partnerships with multiple stakeholders and the convergence of expertise in the life sciences with the physical, chemical, mathematical, and computational engineering and social sciences. One of the benefits of being a member of PAASE is learning from the very diverse and interdisciplinary group of scientists, engineers, and PAASE. And I hope that we can implement team science in tackling the many scientific challenges facing us. Over the last 20 years, we have become very adept in making basic science discoveries. This is especially true soon after the Human Genome Project, where we know more about the nearly 7,000 human diseases that have been discovered, but we are less successful in translating these discoveries into cures and treatments. In fact, there are only about 500 therapies that are available to treat these human conditions. So why is developing therapy so difficult? When we bemoan the cost of prescription drugs or wonder why it takes so long to come up with vaccines and therapies for COVID-19, part of the problem is how drug development has been done for decades. Numerous studies indicate that the average time it takes to develop and bring to market a drug is 10 to 15 years. It also costs on the average about $2.6 billion for every drug that goes to market. So what we're looking at is a current drug discovery paradigm that has a high failure rate of 90%. And this is because the 2D cell culture systems, the animal models that we use currently just do not predict the human condition. And so as a consequence of that, 55% of developed drugs fail due to lack of efficacy. That means it wasn't shown to be effective in treating the human disease. And then 28% fails because of toxic effects in humans. What we need are disruptive technologies. Disruptive technologies in drug development especially that are more efficient and sustainable over the current paradigm. It is because of these challenges that the United States Congress acted in 2011 to establish the National Center for Advancing Translational Sciences. Its sole mission is to catalyze the generation of innovative methods and technologies that will enhance the development, testing, and implementation of diagnostics and therapeutics across a wide range of human diseases and conditions. This shows that instead of actually getting better, we are getting worse in developing new drugs. So that's why I put down there, Houston, we have a problem. And it is actually costing us more. Like in the 1950s, for $1 billion, you can have close to 100 drugs being developed for $1 billion. But as I mentioned, each drug now that goes to market costs on the average $2.6 billion. So there is clearly a diminishing return on investments. So we have poor translation of basic observations into interventions that can directly improve human health. Intervention development process that we use is failure prone, inefficient, and costly. And there is poor or slow adoption of useful tools, resources, and interventions. So my group at NCATS aims to develop, demonstrate, and disseminate disruptive technologies that will change the way we develop treatments and cures. So these are a list of the many programs and initiatives that we have that my group is working on, and I'll be focusing on this particular topic of microphysiological systems for the rest of my talk this evening or this morning. So what exactly are microphysiological systems? These are tissues or organoids on microfabricated chips that consist of multi-channel, three-dimensional, microfluidic cell culture platforms that emulates the activities, the mechanics, and physiological response of entire organs and organ systems. What we aim to do is to represent the 10 major organ systems in the body, such as the circulatory, the endocrine, the GI, immune, skin, musculoskeletal system, nervous, reproductive, respiratory, and urinary system. It essentially means reverse engineering biology into these microfabricated chips. To fabricate chips, we need to take into consideration many things that goes into reverse engineering organ systems into tissue chips, such as the scaffold. What sort of extracellular matrix will you need? What type of cells and the type of cell mixtures, the structure, perfusion, creating microfluidic channels, the bioreactors or chambers where the cells grow, being able to have innervation and an immune system, as well as some built-in readouts, such as microelectronic membranes and computational design. To achieve this will require a diverse group of interdisciplinary experts who are willing to work together and come to a viable solution and make this workable. As an example, if we are to represent the human lung for a tissue chip technology, we have to reduce it down to its basic functional unit, which is the alveolus or the air sac, shown here on this left. This is a rendering of what a lung on a chip would look like. As you can see, there is cyclic vacuuming on each side of the chip that would simulate the negative pressure as we breathe in and out. And so consequently, the cells shown here in blue, the lung epithelial cells are actually being stretched, as well as the blood cells, the vascular endothelial cells in the bottom. So it mimics the biomechanical properties of the lung. Likewise, you can have microvasculature on the chip. So these are tiny blood vessels And so shown here are 2-micron fluorescent beads, similar to what you see for blood cells coursing to these microfluidic channels. You can also innervate, in this case, a gut enteroid, so that it actually undergoes peristalsis, similar to what happens to our GI system after just a nice breakfast or a nice dinner that we may just have had. Because these devices are very small as well, and we need to be quick in assessing the effects of the various drugs. We need to develop real-time biosensing capabilities to tell us what's going on in these chips. So in this particular case, being able to tell when the cells are dying due to toxic effects of a particular drug. For example, fluorescent indicators that turns the cells from red to green to indicate cell death. So those things are also built into the platform. So the tissue chips come in many flavors and sizes. And by flavors and sizes, I don't mean barbecue chips or salt and vinegar chips, but these are actually chips, actual fabricated chips. So this will be the actual lung on a chip. This is the microvasculature on a chip. This will be the GI system on a chip. And for this particular chip, this is actually the female reproductive system. As you are probably aware, females are hardly represented in human clinical trials. And so this is an opportunity to study the effects of, in this particular chip, it contains the vagina, it contains the ovary, uterus, the fallopian tubes, and coupled with liver to inform how the liver is influenced by female hormonal cycle for its drug metabolism. And lastly, our ultimate goal is, of course, to have a 10-organ chip system as shown in this diagram. For predicting toxicity, this is a slide summary showing the many different types of tissue chips that have been developed to examine candidate drugs to see if they have toxic properties. And so we have things like neurons or blood-brain barriers, liver, the GI system. We have human lung, human intestinal organoids, as I've said, the female reproductive system, heart and liver, or heart and liver, and other organ systems. So these are some of the technologies being used and tissue chips being developed to actually test candidate drugs for toxicity. Just to give you an example on how these are used for predicting toxicity. So I'm showing here the liver on a chip, because our liver is the major organ that metabolizes or breaks down drugs. Most of the drugs' toxic effect is manifested in the liver. So in this particular case, Fialuridine is actually an antiviral agent which was discontinued during phase 2 human clinical trial due to liver failure, despite promising preclinical data in rats. So the preclinical data, which was using animal models, showed that it was safe to use in humans. And so in this retrospective study, we tried to recreate why this human clinical trial failed and what was the reason for the failure. So in this particular case, you have rat liver cells on chips, and on the bottom are human liver cells, and they were dosed with 1, 10, and 30 micromolar of Fialuridine. As you can see clearly that the rat cells, when stained with Nile red, still consistently showing the same color, and it's been quantitated here on this chart, whereas the human liver on the chip shows increasing amounts in a dose-dependent manner, accumulation of lipids or fatty deposits leading to steatosis or liver failure in this system. And that is quantitated on this chart, showing that actually the human liver on a chip would have predicted this toxic effect of the drug that the rat animal model or the rat liver on a chip did not predict. And having this kind of data would have saved time and effort in developing this drug, and certainly would have saved many human lives that would have not died because of knowing what the toxic effect would be. Another way of looking at it is to look at albumin secretion, which is an indicator of liver function. And so you can see here that the rat liver on a chip continues to produce albumin, whereas the human liver on a chip, when dosed, again with increasing dose of Fialuridine, starts to show decreasing albumin secretion. Likewise, when you look at markers of liver injury, such as a microRNA called MIR122, which is liver specific in its expression pattern, as well as glutathione S transferase, a specific liver enzyme, and keratin, you can see that these liver injury markers increases in a dose-dependent manner as well, indicating that liver function is compromised and would lead to liver failure. This next slide shows the importance of having interconnected multiple organ systems. Shown here is the schematic of how you could connect a blood-brain barrier, a gut, liver, kidney, with the heart and other control systems. So in this actual study, the liver and kidney were coupled together to study the effect of aristolochic acid metabolism leading to kidney failure or kidney toxicity. So, so aristolochic is a chemical that is found in Chinese herb medicine that is usually taken for weight loss, but unfortunately can lead to kidney failure. So in this particular study, if you just have the kidney on a chip treated with 0 or 5 or 10 micromolars of aristolochic acid, if it's just the kidney cells, the kidney stays happy and alive. But when you couple it with the liver, the presence of just the liver would start to break down the aristolochic acid, and the metabolites actually leads to kidney toxicity, and that's shown in this chart. In order to study what the mechanism is and what that metabolite is, it turns out that it's a sulfated conjugated form of aristolochic acid, and this is shown here in live dead cell staining. So if this is treated without any aristolochic acid, so this is how it looks like. When treated with this conjugated form, this is the dead kidney cells. And interestingly, this sulfated form is actually taken up by the kidney cells using the organic ionic ion transporter 4. So you can actually block the effect or the uptake of this aristolochic acid metabolite by adding 2 millimolar of probenecid, and showing that you can so the mechanism of action in terms of uptake leading to cell death of the kidney cells. So in addition to the use of tissue chips for evaluating drug safety profile, tissue chips are also being developed to model various diseases, such as in this case, Alzheimer's disease, ALS and Parkinson's disease. We have neuropsychiatric disorders, radiation-induced xerostomia. So this is dry salivary gland because of radiation therapy. A lot of rare diseases, modeling cardiac myopathies, atrial fibrillation. You can have kidney diseases, vascular malformations, COPD, and influenza infection in the lung, polycystic ovarian syndrome, and even for complex diseases such as type 2 diabetes with multiple tissue and organ components. So this models, when validated, will be useful for evaluating candidate drugs to demonstrate its effectiveness or efficacy in treating these diseases. So a recent strategy in drug development is repurposing. So in this case, you take an FDA-approved drug and then screen them for effectiveness in other indications other than the disease that the drugs were developed for. So in some ways, taking an already approved drug so that you can shorten the time of development because you already have a safety profile of that drug, and just seeing if it works for another disease or another indication. So in this case, looking at SARS-CoV-2 infection in lung epithelial cells. So in this case, just looking at 2D cell cultures on a dish or in a Petri plate. You can see here shown in the black bar graph, cells that were infected with SARS-CoV-2, and shown in gray are cells infected with a control virus, just to show that the effect is not specific to the particular way that the cells were treated, and would control for non-specific effects. So you can see here that of the 7 drugs that were tested in a 2D cell culture system, for the most part, at increasing a dose up to 5 micromolar, they show quite a dramatic effect in decreasing viral entry. So that means, it might be promising as a preventative to prevent the infection of the lung cells against SARS-CoV-2. But if you were to pursue this study, you would have to test all the 7 candidate drugs, go through the additional follow-up screens, and of course that takes a lot of time and effort. But the value of this 3-dimensional human airway on the chips is that now we can actually see if these are actually viable candidates to pursue. So the same treatment was done at 5 micromolars, and you can see here with the lung on a chip, it's only torimifene and amodiaquine that shows promise in preventing SARS-CoV-2 viral entry. Torimifene is a non-steroidal estrogen receptor antagonist, which could explain too why men are actually more prone to SARS-CoV-2 infection. And then amodiaquine is actually an anti-malarial alternative to chloroquine. So you would note here that chloroquine, while it showed dramatic increase or decrease in viral entry in a 2D cell culture system, actually did not have much effect on the actual 2D cell culture system. And so this is how this system can be used for testing the potential efficacy or effectiveness of a candidate drug. Now, in switching gears and looking at the economics of science, investors usually turn to market indicators, for example, the Gartner plot for promising technologies. New technologies, as shown here, actually go through 5 stages of development. One is the innovation trigger, where certainly the proof of concept or perhaps even a prototype is demonstrated to show some utility. And then, of course, that leads to a lot of excitement and publicity, leading to a peak of inflated expectation. Now, if those inflated expectations are not managed properly, that almost always leads to failures, exposure of the deficiencies in the technologies, and that leads to a number of companies or developers actually bailing out or going out of business. And then those that survive may produce 2nd, 3rd, even 4th generation follow-on technologies with improved SOPs that would then eventually lead to high growth, adoption, and at least a 30% market share. So when we started tissue chips, or the biochips actually was marked here in 2013, down in the bottom, and in 2018, we're right now at the peak of inflated expectation. My goal is to reach the plateau of productivity in as short time as possible. And so the question is, how do you achieve that? What are some of the strategies needed to go about doing that? And so what I have come up with, and in doing the market survey and talking to the scientists and the technology experts, it's clear that the new technology must meet market needs. There should be early engagement with the end users. And then, of course, the technology should demonstrate ease of use and cost efficiency. And so question is, how do you go about doing that? So in the tissue chip program, we have several strategies that we have established or developed to bring this quickly into adoption. So first strategy number one is we establish a validation framework for tissue chips to build confidence in the technology. What does that mean? So it's actually 3 stages of validation. One is physiological. Does the technology itself or the organ on chip actually represent organ function and structure? And this can be demonstrated with a training set of reference compounds where you know what the effects are based on humans, and can these chips actually replicate the findings on the reference compound. So largely this is done by the tissue chip developers themselves. And in fact, we have done this and demonstrated this in a number of publications. So as of 2007, more than 500 publications have resulted from this type of work and this type of validation. The second stage of validation is called the analytical stage. And this is typically done by an independent group of scientists testing for robustness, reproducibility, reliability, and relevance. And this is typically done with a validation set of compounds, biomarkers, and assays. In order to achieve this, NCATS established tissue chip testing centers. And so in this case, we funded MIT and Texas A&M University to be the independent testing centers. And we also created a microphysiological systems database housed at University of Pittsburgh. And finally, the last stage in terms of the validation framework would be industrial. And this is actually where industry, the pharma industry, and regulatory agencies such as the FDA starts taking an interest and starts using them in-house in terms of drug development. And of course, this is done now with proprietary set of compounds and can perhaps done with a CRO, contract research organization type environment. And to achieve this, NCATS encourage these testing centers to actually develop a sustainable revenue generating business model. And so in that regard, MIT actually morphed and evolved into Javelin Biotech, which is now operating as a CRO business model. And then Texas A&M evolved into a tissue chip testing consortium, which is essentially a play for pay model with academia, government, and industry partners. And so using this strategy, we are hoping that it would lead a path to commercialization and adoption. But of course, one thing that we're also pursuing is establishment of a microphysiological system or Tissue Chips International Scientific Conference and professional society starting in 2021. Now, strategy number 2 means identifying and partnering with key stakeholders right from the very beginning, not only for the additional investments, but also for guidance and exchange of information. So this just shows how the program actually evolved through the 8 years or so since NCAT started in 2000. Well, the program actually started in 2012. So we've shown the kind of investments that we put in. So about altogether about $340 million investment through that 8 years or so coming in from the NIH and NCATS. And likewise, we have identified the key stakeholders to engage early, not only DARPA, but also the FDA, which has been a key partner from the start, giving us guidance in terms of how these devices can be approved for regulatory use. So they don't provide any funding, but certainly are key partners for guidance. And then, of course, lastly, the end users, meaning the pharmaceutical industry, representing here by the IQ Consortium. And so currently we have interest coming in from 24 major, worldwide pharmaceutical companies who've expressed an interest in applying tissue chip technology in terms of drug development. Strategy number 3 is being prepared to take on additional partners as interest grows and as potential opportunities come about. So even within partnership within the NIH has grown, as shown here, and different applications of tissue chips from cancer biomimetics I've said to modeling diabetes, to creating immune system on chips, nervous system disorders, blood-brain barriers, biomimetics for infectious diseases. We've also partnered with other agencies, government agencies and funding partners, such as BARDA, the CDC, TRISH, NASA Human Research Program, the EPA, United States Geological Survey, and the Veterans Administration. And certainly have been instrumental in catalyzing the development of technologies, this type of technology in Europe, Asia, and in Australia. Strategy number 4 is encouraging and funding a number of spin-off and startup companies. These are just about 20 or so companies that have already started and formed around organ on chips and organoid on chips and tissue chip technology. So NIH has certainly supported the development of spin-off and startup companies. And this certainly leads to democratization of the technology platforms, allowing pharmaceutical companies and other end users to choose from at least 20 companies for CRO-like services and or purchase of various platforms and accompanying consumables. And finally, we go to Ad Astra, sort of like the cliffhanger, aim for the stars. So Ad Astra, as I mentioned, is Latin, which means aim for the stars. Ad Astra is an admonition that my parents gave their children and grandchildren. On the left is a picture of them when they just got married, and the other was taken in their 40th anniversary. Sadly, they are no longer with us, but I do dedicate this presentation to them since they encouraged me to always think outside the box. And so what does outside of the box thinking mean in this Ad Astra strategy? Well, I think that it's really looking at age-related physiological changes. This is a hard system to model. We all age, just as my parents did, and we will all experience these age-related changes. That includes musculoskeletal changes, such as osteoarthritis, osteoporosis, rheumatoid arthritis, sarcopenia. We can have GI changes, cardiovascular changes, where the heart valves tend to increase in thickness with age, blood pressure tends to go up with age, and you get atherosclerosis and arteriosclerosis. Respiratory changes, renal deficiencies, decline in immune response, and certainly decline in visual acuity or hearing and overall sensory decline. So the question then is, can we model age-related diseases on tissue chips? And can we develop drugs to mitigate or slow down age-related diseases? And who are, of course, the right partners to engage? And so as it turns out, similar aging-related changes happens to astronauts even after only a brief exposure to microgravity. And so for the 800 or so astronauts that have actually gone up in space and spent a considerable amount of time either at the space station, the space shuttle, there are some of the early changes that happen. So within 3 weeks, the early response includes upper body fluid shift, neurovestibular disturbances, sleep disturbances, bone demineralization. Within 3 to 6 months, in addition to above, you have muscle atrophy, cardiovascular deconditioning, GI disturbances, hematological changes, and any astronaut staying for more than 6 months actually suffer much more severe changes, such as declining immunity and renal stone formation. But the major difference between the actual aging that happens on Earth and space-related changes is that when the astronauts return back to Earth, everything reverts back to normal. And so here presents itself an opportunity for us to investigate the biology of aging, during which we can study its development, but also during the recovery, and perhaps identify what those molecular changes are, what those epigenetic changes are, and develop drugs that may slow down the effects of aging for each and everyone here. And so how do we do this? And so again, I consider the Ad Astra as sort of like identifying what the game changers are. So game changer number 1. So what we did was MCATS partnered with NASA and CASES, which is the Center for the Advancement of Science in Space, and the International Space Station National Laboratory for the Tissue Chips in Space program, which is essentially to study the role of the environment, that is microgravity, on human health and disease, and to translate those understandings to improve human health on Earth. And so what we have done here is to look at the effects of aging on the human body and trying to replicate those on the chips. The second game changer, number 2, is the automation and miniaturization required for space flight. And that would essentially lead to broader adoption and use of tissue chips technology here on Earth. And let me illustrate how this works. And so for game changer number 1 is actually our official space patch that would fly on missions, and the various projects that we're pursuing, looking at immunosenescence, the study of aging of the immune system, post-traumatic osteoarthritis, blood-brain barrier permeability changes, immune system and lung infection, looking at the formation of kidney stone and proteinuria in the kidney, some of the cardiac changes and muscle wasting, such as sarcopenia, and the tendency to have gut inflammation and the change in microbiome. As I mentioned, game changer number 2 is looking at technological improvements. And the control systems for tissue chips are actually very complex and quite complicated. And just to illustrate that, so this is sort of like the typical setup in the laboratory to run 24 chips. And so I wouldn't go to all the specifications down here, but just to drive home the point, it's about 48 cubic feet or 1.4 cubic meters. So it's about the size of an average fridge. But as you may know, payload is quite expensive realty in space. And so the demands for this project to go up in space and still maintain the same functionality as it is on Earth is that we have to reduce this fridge-size technology or fridge-size instrumentation to the size of a shoebox. So how do you go about doing that? So again, we partner with NASA, CASES, and International Space Station, and their space implementation partners, TechShot, Space Tango, and Bioserve, and work with the NASA engineers and payload developers towards automation and miniaturization. In just a little over a year since the tissue chips project started in 2017, we were actually quite successful in miniaturizing the technology and sending chips up in space. In fact, this is a picture that I took watching the launch for SpaceX 16 at the Kennedy Space Center, and included in the payload is our immune senescence on chip project. And then in April of 2019, SpaceX 17 launched, and that included 4 projects: the lung infection and bone marrow, proteinuria and kidney stone formation, osteoarthritis and blood-brain barrier permeability. And then SpaceX 20 launched just before COVID-19 lockdown at the Kennedy Space Center. Again, this payload included looking at the aging effects in heart tissues and the immune response in the intestine. So this were, the last 2 were night launches, and just to give you a picture, this is the picture taken of a night launch, equally spectacular as a day launch. And so in such a short period of time, we were able to model aging-related disease. So shown here is actually the Dragon capsule being captured by the space station. And this is one of the astronauts, Christina Cox, working on the tissue chips. And so they spent on the average about 30 days up in the space station. And then they returned to Earth, as we saw return of the Dragon capsule yesterday. And so the samples are retrieved and they're analyzed biologically for omics marker analysis, such as transcriptomics, metabolomics, epigenetic changes, some histology, immunohistochemistry. And then of course, the instrumentation and the platform are examined for technological advances, looking for structural soundness of the chips, platform and instrumentation and experimental automation. What we are planning is that a second flight for all these projects is planned. with the testing of actually candidate therapeutics on those second flight to see if we can actually slow down or mitigate the effects that are seen. So far, our preliminary data shows evidence of aging-like physiological changes just within 30 days in space. So this is compared to current models of aging where it takes months, if not decades, to model aging in other model systems. And so in closing, what I'd like to again, drive home the point that team science is a key enabler to tackle complex challenges and achieve innovative solutions. And that involves partnerships with stakeholders and the convergence of interdisciplinary expertise, as I've shown in this presentation. So far, tissue shifts have touched many areas of drug development, as shown in green. We still have to impact some of the clinical aspects, but we're on our way to doing that with our new program on clinical trials on chips. It's been estimated in 2019 that R&D spending for pharma will grow to $224 billion by 2024. And likewise, a group of industry pharmaceutical experts predicts that tissue chips would save between 10 and 26% of drug development costs within 5 years. And so not only are we able to hopefully be efficient in drug development and make it more economical, but more importantly, we hope to increase the number of therapies available to treat human diseases. And so with that, I'd like to thank you for your attention, and I certainly am grateful for the many partners that we have in this effort, and I'm happy to take any questions.
Thank you very much, Dan, for a very, very interesting talk. And we could see the complexity of how science is done in this team science in this complicated world. And just as an observation, you certainly have reached your stars. That motto ad Astra is very fitting for your talk. And congratulations. So I think I'm going to allow Al Sirafika to ask a question. We'll have maybe 1 or 2 questions before going to the next speaker. Al, you could turn on your microphone and then you could ask your question. Al? So yeah, one of the questions that, so I think it's on now. Al?
Okay, Dan.
You could ask your question. Yeah.
Is it on the chat box? Should I just read it on the chat box?
No, you could ask.
Yeah, Dan. I mean, congratulations, excellent. I listened to your talk on 2016. You've gone space-wise in terms of leaps and bounds.
Thank you.
My question is on the spin-offs that you spoke about.
Yes.
Yeah, on the chat box, I did ask about the 20 spin-offs. How many of them received SBIR? I did get the SBIR from NIH in '97. So I'm curious how many of them on the spin-offs from NCATS have gotten that kind of support. And what kind of arrangements do you normally give out for the spin-off?
I actually don't have the numbers at the tip of my fingers, but I'd say we've sustained and supported probably about anywhere from 1/3 to about 50% of them. A number of these companies are actually foreign-based. They started forming companies in Europe. And of course, as you know, with SBIR, STTR rules, that only goes to US-based companies. So a number of companies have actually, at least I'm aware of 2 or 3 companies based in Europe that have actually started operations in the US so they can get SBIR, STTR grants from the United States. And so they range anywhere from phase 1, phase 2 support, anywhere from a typical $175 to $1.7 million for a phase 2 award. And most of them are actually now commercializing these chips, not only the platforms themselves and the cells, but also offering a fee for service as part of their business model.
Okay. And last question: Have you done anything for Asia so far?
I've certainly traveled extensively to Japan, to China, Singapore, Korea. I'd say, and primarily because they had invited me to find out about the program and how they could start similar programs within those countries. So far, Japan with AMED have been successful in launching their tissue chip program about 2 years ago. They're in the process of developing 4 tissue chips, and China likewise is following closely at the heels of Japan. Korea is still in the process of developing this technology. So certainly within countries other than the United States, I'd say Europe is probably close to the technology as the US is.
Okay, one more question, and this is from Lisa Birata. She's wondering if,
Hi, Lisa.
thank you, Al. Yeah, she's wondering if there are any drugs that had been approved by the FDA based on data using these tissue chips.
Not yet. That's
Okay.
actually my dream and hope, is that we actually take a particular drug and test it on chips and take it all the way to IND submissions just based on pure chip data. What so far has been going on is that the chip data are being used, supplemental data to the conventional or the traditional way of preclinical development. And that is because the FDA, I know Lisa works for the FDA, is primarily requiring companies to have certain guidance in terms of how to develop preclinical data, and they require 2 animal species, and so that is still a requirement. Although recently the FDA have actually started paying attention to this technology and have now formed what's called an alternatives methods working group within the FDA to start looking at this technology and how it could be implemented in drug development. So I'm hoping that soon that will happen.
Yeah, thank you, Dan. Gisela has a question, I think, and then we'll proceed to the next speaker. Gisela?
Congratulations, Dan, for your out of a box, out of this world space research. I'd like to ask in particular about your respiratory model for SARS-CoV-2 infection. What stage of infection was it? Early stage, middle, or late severe stage? Secondly, in relation to the controversial hydroxychloroquine, did you test it with and without zinc? Thanks.
Yeah. Good question, Giselle. Thank you. So for the first question, the testing was done on early stage of infection. So what we're trying to do is actually looking and screening for drugs that would prevent viral entry. So in some ways, preventative in some ways, or mitigating the disease from getting worse. So early stage of infection. In terms of the second question, how we tried hydroxychloroquine and azithromycin or zinc, that is work that is still ongoing right now in terms of testing. And as you know, since the original description of this being purely respiratory back in March, the number of pathologies has actually grown and increased. Doesn't just affect the lungs, it affects the kidney, affects the heart, affects the brain, and more especially the blood vessels, causing embolism. So all of those are now active areas of investigation using these chips.
Okay, one final question, and Joel promises that it's a short one. Joel?
Yes, thank you. Dan, congratulations, excellent talk. You referred to confidence building for the adoption of this technology of organoids or tissue or cell on a chip. And I was kind of thinking that perhaps one way to foster that and also to ensure the efficacy of the chip for the test of the drugs is standardization, particularly of the organoids, defining it as a kind of a functional physiological unit for any particular organ. I was wondering whether your center has thought about doing that or implementing that or coordinating that with the stakeholders, coming up with standards for these organoids?
Yeah, so that's a very excellent point, Joel, and you're certainly right on the mark. Since this involves many disciplines, as it turned out, working with engineers and on one side, the physical scientists, and on the other side, the biologists or the life sciences, it turns out that, of course, as you know, as you would predict, the engineering aspects of things, you can engineer, microfabricate endlessly and come up precisely with what you want. But life or the life sciences is complicated, especially the biology. We have to consider diversity, we have to consider gender, we have to consider culture conditions, the sources of the cells. And so all of those are now certainly undergoing standardization. Do you use primary cells, which is taken directly from patients, or do you use stem cells? When you use stem cells, do you differentiate them in a particular way? So not only the sampling, the extraction, and the differentiation, all of those have to be standardized. But that is in some ways, it's an area that goes just beyond this field of tissue chip technology. That is a common pervasive problem within the area of stem cell biology, cell biology, and biorepositories. So we're trying to make an impact as it relates to tissue chip technology, but you hit upon a pervasive problem in biology.
Okay, so thank you, Dan, and thank you for an excellent presentation, very informative. And,
thank you.
And of course, people could still ask questions later on the final question and answer, and they could ask Dan some questions. So let me introduce